Spinal cord injuries are just one of the most devastating things that can happen to a person, often leading to permanent paralysis. For decades, finding a real, effective treatment has felt like it's just around the corner, but always out of reach. But what if the solution isn't some single magic bullet drug? What if it's more like a microscopic special ops team, engineered to carry out a super complex repair mission from inside the body?
I mean, really think about that. What if we could literally design and send in a tiny biological repair crew? A team that could navigate through the bloodstream, find the exact point of injury, and then carry out a whole multi-step plan to clean up the damage, calm everything down, and start rebuilding what was broken. It sounds a lot like science fiction, I know, but it's actually the core idea of a really groundbreaking new study.
So to get how this new solution works, you first have to appreciate just how massive the problem is. See, after a spinal cord injury, the site isn't just a simple break. It becomes a biological war zone. There's this flood of inflammation that creates a really hostile environment, and the body's own defenses build up this barrier that, yeah, it's protective, but it also walls off the injury, keeping pretty much every potential medicine out.
And that right there brings us to the first huge hurdle for any kind of therapy. If you want to fix the spinal cord, you've got to be able to get to it first. And that means you have to get past its very impressive fortress. This fortress has a name. It's the blood-spinal cord barrier, or BSCB for short. You can basically think of it as the spinal cord's own elite security system. It's this shield of cells all packed together super tightly, and it's amazing at keeping toxins and bad stuff out of the nervous system. The problem is, it's so good at its job that it also blocks something like 98% of potential drugs from ever reaching their target. So, how in the world do you get past the best security system there is? Well, you don't just try to smash down the gates with brute force. You get smart. You design an elite team that has an all-access pass. You engineer a smarter therapy.
For a mission like this, you need a vehicle that's super small, stealthy, and biological, something the body's going to recognize as friendly. And the perfect candidate for the job? It's called an extracellular vesicle, or an EV. These are these tiny, nano-sized packages that our cells naturally use to talk to each other. They're like the body's own internal postal service.
And the way they engineered these things is just brilliant. It's a three-step process. First, they picked the right source. They took these vesicles from special immune cells called M2 microglia. These are kind of the peacekeepers of the nervous system, known for promoting repair. Second, they gave them a GPS. They attached a little homing beacon called angiopep2, which is designed to lock onto a receptor that's found right at the injury site. And third, they gave them a supercharge. They pre-treated the cells they came from with melatonin, which has these really powerful anti-inflammatory effects.
And here is the absolute proof that it worked. Now, don't worry about all the data here. Just look at the images on the right. Those are scans of mice. And that glowing red stuff, those are the vesicles they injected. Look at how that bright red signal is perfectly concentrated right where the spinal cord was injured. It's a direct hit. The vesicles didn't just float around randomly. They went exactly where they were programmed to go. Okay, so our special ops team has made it past the defenses and is at the target zone. So what's the plan now? Well, it's not just one thing. It's a whole coordinated, multi-pronged attack on the injury itself. First up, cleanup duty. After an injury, the protective coating around nerve fibers, it's called myelin, it gets all shredded, and that creates this toxic debris that actually stops healing in its tracks. So check out the image here. The engineered vesicles basically encourage the local cleanup crew, those are the green cells, to get way better at gobbling up all that nasty red debris. All right, with the junk cleared out, it's time to rebuild. And that is exactly what these incredible microscope images are showing us. If you look at the images from the treated group, you can see new, healthy layers of that myelin sheath re-wrapping around the nerve fibers. So the vesicles are actively kickstarting the reconstruction of this super important insulation.
Next, they play the role of diplomat. The injury site is just swarming with these angry pro-inflammatory immune cells. You can think of them as being in attack mode. What these vesicles do is they help convince those cells to switch from that destructive mode to a helpful anti-inflammatory repair mode. They're basically quieting down all the biological chaos.
And this might be the most elegant part of the whole mission. The vesicles don't just sneak through the fortress and leave the door open behind them. No, they actually help rebuild the walls of the fortress. This image shows them helping to restore those tight connections between the barrier cells, which makes the barrier less leaky and stops more inflammatory cells from getting inside. So the mission was already a huge success. But it turns out it had this completely unexpected and incredibly beneficial side effect. The team's work wasn't just limited to the spinal cord. It turns out that angiopep2 homing beacon doesn't just target the spinal cord barrier, it also targets the blood-brain barrier. So as these little vesicles were traveling through the bloodstream, some of them took a detour and made their way right into the brain. They found a secondary, totally unplanned target.
So why does this matter? Well, we now know that a spinal cord injury can set off chronic inflammation in the brain, which leads to things like cognitive problems and depression. But this therapy helped with that too. This slide shows the difference. On the left, in the untreated brains, the immune cells are all balled up and angry. But on the right, in the treated brains, the inflammation is way down, and the cells are back to their healthy, normal shape. The therapy wasn't just healing the spine, it was healing the brain's secondary injury too.
So, let's get to the bottom line. Did all of this amazing microscopic work, the cleaning, the rebuilding, the calming, even the unexpected brain boost, did it actually translate into real, measurable recovery?
The answer is a huge, definitive yes. This chart shows a standard score for motor function in mice. The control group in the first bar shows almost no recovery. The group that got the regular vesicles in the middle did a bit better. But the group on the far right, the one that got the fully engineered melatonin supercharged vesicles, they show by far the biggest improvement in motor function. It's not even close.
And this right here, this is what that bar chart actually looks like in the real world. This is a special gait analysis of a treated mouse walking. And you can see, unlike the untreated mice, it's actually able to support its back paws and walk with a much more regular, coordinated rhythm. This isn't just a number on a chart, it's visible, functional recovery.
The researchers who came up with this incredible mission, they summed it up best themselves. They said, "This approach represents a promising new therapeutic strategy for spinal cord injury." And that's a really powerful statement because it validates a totally new way of thinking about this problem.
Which, of course, brings us to one last, really exciting thought. We just saw a targeted, multi-talented, microscopic repair crew successfully treat one of the most complex injuries you can imagine, and it even had bonus benefits for the brain. So the big question this leaves us with is, could this same special ops blueprint, using these targeted, supercharged vesicles, be the key to unlocking real repair for other devastating neurological conditions? Things like stroke, Parkinson's, or maybe even Alzheimer's disease.